Yeah, good morning, everyone. My name is Ryan McClure. I'm a researcher here at PMNL, looking at a lot of different biological systems and kind of interactions between species. So today, the focus is mainly on data visualization and the statistical analysis. So you've already gotten a good introduction on how this data is kind of processed after it comes off of these machines. But after it's processed, how do we take steps to sort of interpret that data? So to kind of get things kicked off for today, we are going to hear from Susannah Tringe. She's going to give a really interesting talk looking at microbial drivers of methane emissions. Susannah is the division director for environmental genomics and systems biology at Lawrence Berkeley National Laboratory. Her research is really focused on how microbes tend to influence greenhouse gas uptake and release in wetlands and in agricultural systems. And in addition, how microbes affect plant growth, plant health, and stress resistance. So Susannah, I've got your slides pulled up here. So if you just tell me when to advance, we should be good to go.
So as Brian said, I'm the division director for environmental genomics and systems biology, but most of my time at Berkeley Lab was actually at the Department of Energy Joint Genome Institute, which is another user facility funded by the DOE BER to support genomic work in the user community, similar to EMSL, and just has very complementary capabilities. And so the project I'm talking about today has actually used a lot of JGI resources as well as some EMSL resources. All right, next slide. So today I just put up a quick outline. I'm going to tell you a little bit about the kind of motivation, which includes, carbon cycling and wetland restoration and methane and microbial communities in our local area, the San Francisco Bay Delta. And then proof of principle project that we did on an island called Twitchell Island several years ago now. And then also a project studying the South Bay salt ponds. And I give a little teaser about what we were able to find in each of those. All right, next, maybe 2 slides. All right, so, everyone here, I'm sure, is already familiar with the carbon cycle and global warming. Why don't you advance another, Ryan? This is just kind of a reminder that we're putting too much carbon into the atmosphere, and certainly that needs to be reduced. But one other strategy, which I guess has gotten a lot more publicity lately, but was less talked about, when we started this project, is that we also want to get carbon out of the atmosphere, and we really need to look at all the options we have available to us for quickly reducing carbon that's in the atmosphere. And actually, quite a lot more carbon is in the soil, which I'm sure you've already heard about this week. So that anything we can do to tip the balance in favor of carbon being not in the atmosphere, that's fine. You can go ahead and advance. And then, I just have a little slide here is saying that, that this idea has been proposed in some of these carbon offsets, which sometimes if you pay for a carbon offset, it pays for, something that's just reducing carbon going into the atmosphere, like renewable energy. But there are also these programs to remove carbon from the atmosphere, such as planting trees. And this is just showing that California has a carbon market that does give credit for forest reforestation. But in the next slide, you'll see that actually wetlands, specifically like coastal vegetation systems, have even greater potential for storing carbon in terms of the rate of carbon accumulation and the amount of carbon storage per unit area, and the persistence of that carbon actually has a lot of favorable features in this coastal vegetation. So this is called blue carbon sometimes, and that's a promising avenue for taking carbon out of the atmosphere. All right, next slide. And so a little bit about wetland restoration. You can also advance, I think, 2 or 3. I've got a couple of animations on there. So wetland restoration is carried out for a variety of reasons, because it can restore habitat for all variety of wildlife, sometimes endangered wildlife. It can improve water quality. But our real focus here has been carbon, because wetlands store a lot more carbon relative to their global land area than other systems. So wetlands make up, less than 10% of the global land area, but store something like 1/3 of the total carbon. And so the reason for that, click through maybe 2 or 3 more animations is that wetland vegetation takes up a lot of carbon dioxide through photosynthesis. Then that carbon ends up in the soil where it tends to break down very slowly in the anoxic conditions that persist in heavily waterlogged soils. But unfortunately, that can also lead to the production of methane, which is a much more potent gas, the greenhouse gas, than carbon dioxide. And therefore, even if you were sequestering a lot of carbon through photosynthesis, if even a small fraction of it were to escape back into the atmosphere as methane, you could have still a negative net greenhouse gas effect. So it's very important to take that into account if you're trying to store carbon in wetlands. All right, you can go on to the next slide. There'll be a couple more about methane there. Yeah. So our big question in this project was just, well, what major factors determine if a wetland serves as a greenhouse gas source or a greenhouse gas sink, with a particular focus on the microbes, which are the ones that break down that organic matter and then ultimately convert it to soil carbon or to methane or to carbon dioxide that end up back out in the atmosphere. All right, next slide. And so our study system is the local San Francisco Bay Area. And so you can click a couple more. So that in the past, the Bay Delta region, sorry, well, maybe go back to the previous slide, but that was ringed with a whole network of tidal wetlands. You can see on the left, that green area, all of that was wetlands. But most of those wetlands have been drained and converted to other uses, in particular farming up in the Delta and commercial salt production in the South Bay. And so I'm going to talk about restoration projects that are addressing both of those systems. All right, so then the next slide is just showing that there are these kinds of restoration projects going on all over the Bay Area and really all over the world. Most heavily motivated by things like wildlife habitat and flood control. And so one of our motivations has really been, especially if you're already going to restore the wetlands, can you do it in a way that is most favorable to carbon, which hasn't been the primary motivation between a lot of these wetland restorations, although the first one I'm going to talk about was very much about carbon storage. So next slide. So that was this on this island called Twitchell Island, and you can go on to the next slide. And that was in the Delta, where there's a unique environment where these there are these islands in the Delta. It's really quite freshwater because it's water coming in from the Sacramento and San Joaquin rivers, which is freshwater, but it is actually close enough to the bay that it has some tidal influence. But these islands have made great farmland because they have these very rich organic soils. And so over the over 100 years ago, most of them were diked off, as shown in the next, the second tier here, and then used for agriculture. And sorry, these soils then tend to start to decay because once they're drained and exposed to the air, they decompose. And then that creates this very vulnerable system where the land surface is far below the surrounding water. And when the levees fail, which does happen, as shown in the next slide, you end up with effectively just open water. It's too deep to get back to that wetland state that it started out at. And so this is just an image of a levee failure that happened. And on the right is just a picture attempting to show, it doesn't show all that well, but when you're there, it's really quite striking. When you're on the levee, which is where the roads are, the water can be right next to you, and yet the farmland is meters below you. So it's really prone to problems. And there have been places where these levee breaches have happened and they have not tried to reclaim the island. It was just too dramatically flooded. And if this happened on a large scale, it could even result in saline water influx from the bay, which could affect, the drinking water for the region and the farmland and have lots of negative effects. And so there's an immediate threat related to this land subsidence, and then, but also a potential carbon opportunity, which is what is shown in the next slide, that scientists from the USGS and other organizations created this restored wetland to see if they could develop a strategy for actually building back that soil carbon. And so next slide, or next animation, they sort of penned off a little part of the island and flooded it very shallowly at either 25 or 55 centimeters water depth. And we're able to achieve very good vegetation and very good peat accretion of as much as 4 centimeters a year, and a net greenhouse gas budget that was negative in storing quite a bit of carbon under the soil. So our question, let's see, click the next arrow, I forget what comes up next here, was, what is the microbial contribution? And so we sampled different parts of this wetland where the level of peat accretion was different because it wasn't uniform throughout the wetland. There are places that still only grew a little bit of vegetation, places that grew a lot more, which tended to be related to the water flow. So if there was a lot of water flow near the water inlet, the peat didn't accrete as fast as the sort of more backwater. When we sampled and looked at the biochemistry, we found that electron acceptors like oxygen, nitrate, and sulfate tended to be highest at the water inlet and more depleted farther away. And then one animation forward, the measurements of methane that we made with our colleagues indicated that that was an inverse gradient, that the methane fluxes were highest in those sort of backwaters where there was high peat accretion, which is not surprising and is consistent with what others see, although it does mean that you can't simultaneously optimize for high peat accretion and low methane flux, that they're very correlated with each other. So you kind of have to find some balance between the two. All right, next slide. Or then the animation is just, our question at the time, which seems maybe naive now, when we started this project, was like more than 10 years ago, was just, can you actually detect changes in the microbial communities that relate to these changes in geochemistry and use them to interpret what's happening in the system? Our previous studies in metagenomics before this had been comparing very disparate systems, demonstrating you see different complements of genes in like ocean versus land. But we weren't sure if we could really detect differences in gene abundance or microbial communities in like one part of a wetland versus another part. All right, next slide. And in particular, we're really interested in this methane question, both because methane is such an important greenhouse gas and because methanogens are actually a fairly well-defined microbial group. So they made a good target for a study because we knew we could probably recognize the organisms and the genes involved in methanogenesis in our data. And so they would be a good proving ground for these approaches. And so there are a couple of different hypotheses on like why methane might be different in different parts of the wetlands. And the first was just that there might be, more methanogens where there's more methane. You can click through, there's like probably 5 animations here. Another would be just that the methanogens become more active under certain conditions. And then another would be that there's actually different methanogens that prefer different locations and that might have different levels of inherent methanogenesis capability. All right, you can go on to the next slide. But in order to do any of this, we needed to be able to actually measure methane. And a great thing about this Twitchell Island site was that they had these eddy covariance towers already there, and they were studying greenhouse gas budget. Particular a group at UC Berkeley led by Dennis Baldacci has been super helpful, and they have done some really in-depth studies of the delta and the potential for carbon sequestration there. So this is great for the whole system, but the eddy covariance towers collect greenhouse gas data over very large footprint based on the wind patterns. And so they're great for getting a budget for the whole wetland, but not useful for looking at microbial level differences in different parts of the wetland. And so for this study, we also did, if you go through, click through maybe 2 more, our colleagues at USGS helped us with doing some microcosms, taking samples back to the lab and seeing how much methane they produce. They also did these in situ chambers, which is a standard method, although not as easy to do when you have vegetation that's, meters tall. And so they had to create these kind of structures and mylar bags, you can see in the lower corner, to kind of bag the vegetation where a lot of the methane comes out through the plants, through the parenchyma, and then measure the greenhouse gas production within those chambers. So that was what those 2 methods are kind of what we used to determine in this first wetland where the high versus low methane sites were. And that was the level at which we were able to do it then. Since then, we've started using this Los Gatos Research Greenhouse Gas Analyzer, which is great because we can actually take a soil core and put it in this jar attached to the machine and then immediately measure methane production, ideally right in the field, which is what this picture is showing. I think there's one more animation there. And then we can take that core and extract DNA from it or metabolites or whatever we want to study and do that on exactly the same sample as we did the methane measurements. So you can really be sure that we're not introducing an artifact by looking at different samples for those different measurements. All right, so next slide. And so when we started studying the microbial communities at Twitchell, we started out doing standard 16S amplicom sequencing, which I'm assuming someone's probably already covered this week since we're on Thursday. And we found that these communities were strongly patterned by site, which is associated with methane emissions. You can see these 3 different sites, the sort of close to the inlet and the far away from the inlet and the intermediate kind of pattern, just like that. And then 2 more animations. The organisms that we saw that differentiated these sites made a lot of sense. And so like the lower methane site had a lot more sulfate reducers and nitrate reducers. This is just one example of a Desulfobacteriaceae. And there were some methanogens that were more abundant at the high methane sites. And so that was encouraging for there being detectable differences in the microbial community. But what we really wanted to look at was functional genes, which is in the next slide. And we also saw really strong patterns in gene abundances. Maybe click through to, I think there might be 4 more animations there. And we found, so in these higher methane sites, there were distinctly more methanogenesis genes, and that's shown. All the gene names that are in red are associated with methanogenesis, and then these other colors that I have in the words are, so the blue and the green are metal reduction and denitrification, dissimilatory sulfate reduction. And there's kind of a checkerboard pattern. If there were a lot of methanogenesis genes, there were a lot fewer genes for those other anaerobic metabolism genes. And so again, that was consistent with these patterns that we were seeing in methane production. Interestingly, the methane oxidation genes kind of showed a different pattern, and they tended to be more abundant in samples that were taken close to the plant roots. So we actually took bulk soil samples and then sort of more rhizosphere samples. And that is also expected because the plants are both a conduit of methane out of the soils and a way of introducing oxygen down into the soils. So at the plant roots is someplace where you expect to see both methane and oxygen to allow methane oxidation to take place. All right, next slide. And then finally, I mentioned that we thought, methanogens might be more abundant. We also thought that there might be, the activity was something weren't really able to do a good job of looking at. We did look at gene expression, which was not dramatically increased within the higher methane sites versus the low methane sites, at least not any more than the gene abundances already were. But we did see differences in the identity of the methanogens there. And so maybe click through, I think probably 4 more animations there. And so if you look at this tree of methanogens, which I realize is very illegible, but all you need to see is the colors. The colors are still the same ones I've been using throughout the talk, where the red is the high methane, green is intermediate, blue is low. And there are some branches on this tree that have methanogens from all of the sites, so they're kind of widespread. But then there are some that seem to be kind of specialized. Or do I have those? Let's see. I guess I stated my red and green and blue backwards. Sorry, but there are some methanogens that seem to be associated with the lower methane sites, and then others that were associated with the higher methane sites. And if you click twice more, those actually showed different mechanisms of methanogenesis. And so the hydrogenotrophic methanogens were the ones that were associated more with the high methane, and then acetoclastic methanogens were more associated with low methane. And so if you look at the next slide, that just shows kind of the overall methanogenesis pathways. This starts telling us a little bit about how that methane might be getting generated in these sites, so that we can learn a little more about what's driving those patterns. So that's actually, so that this study was kind of our, like I said, our proof of principle that was published back in 2015, I think. And since then, we've moved on to some other sites. So if you go on to the next slide, we've actually done a study of the whole kind of Bay Delta region that I'm not going to talk about, but then a nice example that was one we've worked with EMSL on is these South Bay salt ponds. So if you go to the next slide, just to lead in with a little bit of data from our cross Bay Delta study, we did have sampled microbial communities and measured methane emissions at both restored and historic sites across the salinity gradient. So all the way from fresh water to even a little bit more saline than seawater. And what you expect to see along that kind of gradient is a gradual decrease in methane emissions. And that's basically what we saw as kind of a log linear, because seawater has a lot of sulfate, and sulfate reduction is generally more energetically favorable than methanogenesis. So you expect sulfate reducers to outcompete methanogens as you get more sulfate present. If you go to the next slide, in these South Bay salt ponds, we sampled both restored and unrestored salt ponds that were very hypersaline. And our initial expectation was that these hypersaline sites would actually also have very low methane production. But we were interested in looking at them as a model for restoration, because there were these examples of that was a, well, I'll show the next, I think it shows in the next slide. I'll go on to that one and then come back to this. There is this big project called the South Bay Salt Pond Restoration Project, where Cargill, the company that was producing salt at these sites, turned over a large area of these salt ponds that had been used for industrial salt making to a nonprofit for restoration. And I should confirm this, I believe it was part of a deal to expand the San Francisco airport. There was kind of remediation going on in one place while there was sort of some more damage being done in other places. But it created this great project to restore a lot of the bay to a more natural state. But it's been taking place over a very long period of time, because again, it's the kind of system you can't just open everything up, or you would cause a lot of downstream harm, that these salt ponds aren't really ready for just tidal introduction, but they're carefully designing these wetlands and flooding them over time. And there's a couple of pictures down in the lower corner. So from above, you might have seen a view like this before if you've ever flown into San Francisco, because there are these very brightly colored ponds, which are these former industrial salt making ponds. They're bright red from the pigments of the microbes that inhabit them. And then these pictures on the lower right, the one on the left is actually a picture of us out at sampling of one of the unrestored ponds. And you can see the salt crust going around the edge and the red water in the middle. And on the right is one of the restored ponds, where they actually build these little nesting islands for the birds, because there's a lot of problems with predation of the eggs. And if they can put the nests on islands, they're less likely to get eaten by raccoons or whatever other land animals that might be eating them. All right, the next slide. And so, again, like I said, this is sort of a compression of all the data that we had from across the bay. Our expectation, if you click again, was that, you should, as you go up in salinity, continue to go down in methane production. But if you click through maybe another animation or two, then when we actually sampled these hypersaline ponds, we actually had quite high methane production. And maybe click one more time. And these are just pictures of those systems. And so those unrestored ponds are the ones in the triangles that have generally pretty high methane emissions, which was really unexpected based on the known energetics of these systems. The restored ponds were essentially restored to normal salinity, but still had slightly elevated methanogenesis. And then a nearby tidal salt marsh, which had never been used for salt making, had very low methane emissions, and in fact, sometimes methane consumption. All right, next slide. And then just replotting that. And so this is shown in the context of our bigger studies, this points in the Delta and the Bay, and then all of our samples in the South Bay. We actually got this interesting pattern of, in the Delta, like I said, we did see an interesting pattern in the Delta where there were some slightly higher methane emissions at very slightly saline sites, but then, the methanogenesis went to essentially zero at the more seawater salinity, but then really ramped back up in these hypersaline sites, which was completely counter to what we would have predicted. Next slide. And so, next we kind of looked at a few different things. Here's just a plot of the data showing, if you plot the methane flux from each of these systems, it's super high, on average from the unrestored. It actually correlates with salinity rather than anti-correlating with salinity. And the sulfate is also very high too. So that was one question that we had was, well, we know the salinity is really high, but, could the sulfate actually not be that high? Could it have been consumed while these were separated out from the seawater for, decades? But in fact, this sulfate is very high too. So that was not an explanation for the low or for the high methanogenesis. Next slide. However, then when we looked at the literature, we found a potential explanation in that others had seen methanogenesis in hypersaline sites that was attributable to a third type of methanogen that uses methyl-containing substrates. So I earlier mentioned the acetoclastic and the hydrogenotropic methanogens, but some methanogens use methyl-containing substrates such as methanol or methylamine, and there's actually several others that are now known to potentially feed into the methanogenesis pathway. And that those substrates are not accessible to sulfate reducers, and therefore you can actually have sulfate reduction and methanogenesis going on at the same time. And my postdoc Susie set up experiments to test this by adding these methyl-containing substrates, and found that actually we got pretty good methane production from trimethylamine, but not from acetate, and then a little bit from methanol, but not all that much. And so that seemed to confirm that it was these methylotropic methanogens that were likely to be causing the methanogenesis. But we were not readily able to confirm this from 16S data, because our 16S data actually had very few methanogens in it at all. And we had started it out as a metagenomics project. So in parallel to all this work, we also generated a lot of metagenome data, which I think I start showing on the next slide. And so this is the 16S data, just showing some of our data. The diversity was dramatically decreased in hypersaline, which we kind of expected since it's such an extreme condition. The restored and the historic were certainly not the same. The restoration had not brought the system back to the same microbial communities as in the sort of unaffected tidal salt marsh, but did kind of become more similar to those pre-salt making conditions. And then this is just showing, looking at a guild level, there were these really pretty low levels of methanogens, but still more abundant in the unrestored. And then there's, a couple of other guilds shown there in terms of things like sulfate reducers, which are abundant actually throughout the ponds, but much more abundant in the restored and the reference than in the unrestored. All right, and then the next slide. But then when we generated the metagenome data, we were expecting to basically confirm this hypothesis that it was methylotropic methanogens, but we generated all the data, and then we annotated all the data, and we looked at the relative abundance of just genes in the system. So without breaking things down into individual genomes, which I will show in a minute, we assembled and annotated and then compared gene abundances among the different samples, and then looked at the genes whose abundance specifically correlated with methane production from the same core that we had collected at the same time. And the archaeal methanogenesis genes were very strongly correlated with methane, and that's shown in that top set of genes on the heat map. But another set of genes that that came out of that analysis as being very highly correlated with methanogenesis was these genes for degradation of methylphosphonate. In addition, we saw some anti-correlation of methane with genes for methane and or ammonia oxidation. All right, if we click through one animation. So these genes for methylphosphonate degradation are also an interesting story, which has come out in the last decade or so, that these methylphosphonate compounds contain a direct carbon to phosphorus bond. And when you cleave that bond, it actually releases methane into the atmosphere. So it's a mechanism by which microbes can produce methane that is completely unrelated to archaeal methanogenesis, the sort of known pathway of methane production from microbes. And it has actually been demonstrated to be a major source of methane in the ocean, particularly in sites that have been observed to produce methane despite being not anaerobic, being oxygen containing. So if you should click through next animation, and it's especially likely to be active in environments that are phosphorus limited, because these methylphosphonates are difficult to break down, but they are a source of phosphorus. And so if there is very limited phosphorus, organisms will, in a sense, be motivated to break these down to get the phosphorus from it. And then the methane is really just kind of a byproduct. It doesn't benefit the organism because it just goes out into the atmosphere. And if you look at the nutrient content of our salt ponds, and specifically the nitrogen to phosphorus ratio, which should indicate to some extent how limiting phosphorus is, because you would expect those to be at a certain ratio, which I don't remember the number exactly. But in any case, the salt ponds are at something like 10,000 to 1 for nitrogen to phosphorus. So meaning that phosphorus is very, very limiting in these environments. Again, suggesting that methylphosphonate breakdown would be a favorable process for the organisms there. All right, next slide. Just looking a little deeper at the genes and the genomes associated with methane flux. So we also took these metagenomic data and assembled and then binned into genomes, or metagenome assembled genomes, or MAGs, and then looked at those at the abundance of those genomes and the genes for these various processes. So I think, sorry, I'm looking at this on my phone, but these at the top is just showing the correlations, I think, of just the gene level, the abundances with methane, showing that methanogenesis and methylphosphonate degradation correlate well with methane flux, and the methane and ammonia oxidation are anti-correlated. But then at the bottom is a tree of the metagenome assembled genomes that we derived from the salt ponds and the restored systems, demonstrating that there was really only one methanogen that assembled effectively out of the metagenome data, and it did contain all the genes for methyllotrophic methanogenesis. But there were actually many genomes that assembled out of the salt ponds that contained all the necessary genes for methylphosphonate degradation. And those genes were also much more abundant in those unrestored ponds. All right, next slide. So our hypothesis based on this is basically that there are contributions from both methylotrophic methanogens and methylphosphonate breakdown. This is just showing we've gone a little deeper and actually extracted these MAGs. So here's the methanogen MAG, which was a novel genus that we've named Methanogenesis. If you click through again, this will just shows that one more, that genome does contain a complete pathway for methylphosphonate degradation, or sorry, not methylphosphonate, for archaeal methyllotrophic methanogenesis, as do many of its neighboring organisms have similar pathways. And then in the next slide is just 2 MAGs that contain all the genes for the methylphosphonate degradation. You can click maybe 2 more times and show where those fall on the tree. Those were more related to this genus Meravita. And I think they were more likely to contain these genes for methylphosphonate degradation. Not all of their close relatives actually did. So those seem to be genes that may be a little more mobile. So then in the next slide, what we then proposed to EMSL was to try to dive in a little deeper and see if we could learn a little more about the mechanisms of methanogenesis. And in particular, what we'd like to do is model this system and see what kind of changes might modulate that methane production, with an eye to being able to predict what happens when you restore these salt ponds and how you could minimize methane production. And so we've been working with K-Base to actually pull together all these potential pathways for methane production, which aren't necessarily currently all represented in standard sort of metabolic maps, and then try to model the methane production. And at the same time, we've worked with EMSL to generate some metabolite data so that we can get some kind of anchor points on those models. So the next slide, we've gotten some great metabolite profiles for these samples, especially for the unrestored and tidal salt marsh sites. Unfortunately, for some reason, the restored wetlands were very non-amenable to the metabolite profiling. And so we got one taunting sample output, which you can see in the green on this heat map, has a very different metabolite profile from either the historic wetland or the unrestored sites. But we did see some strong patterns in metabolite abundance that were different, not surprisingly, between the restored and between the unrestored and the native salt marsh. I think if you click again, a couple of the really abundant chemicals were trehalose and betaine, both of which are known compatible solutes, which is the chemicals that microbes produce to help adapt to highly saline conditions. So they're sort of osmoregulators that they accumulate in their cytoplasm that help them maintain balance without having an influx of that hypersaline material. So next slide, we've looked at the abundance of these metabolites and how it correlates with methane production. I think we Again, trihalose, this compatible solute, and also acetate were very highly correlated with methane flux. A number of other carbon substrates were negatively correlated with methane flux, sort of giving us a few more hints of where these things might be working together on the metabolic map. And then I think I have one more slide, and that's just highlighting the trihalose there and the acetate. And yeah, you can go on to the next. And then we've also done some additional substrate addition experiments where we added a couple of the methylphosphonates, specifically methylphosphonic acid and dimethylmethylphosphonate. And we've seen moderate methane production from methylphosphonic acid, not so much from the dimethylmethylamine compared to control. And so it's supportive of there being some methanogenesis from methylphosphonate, but not super dramatic because we actually still see pretty high methane production just in the control. One thing we haven't been very successful, despite hard work at EMSL, is in actually detecting methylphosphonates in our system. So we're still a bit up in the air on whether there's sufficient methylphosphonates there to fuel the methanogenesis. So I'd say the data from EMSL is kind of tipping us back toward our original thought that it may be, despite their low abundance, those archaeal methyllotrophic methanogens may be really the major methane producers in our system, but we're still really trying to dig in deeper and be sure of that. All right, next slide. Let's see, where are we on time? It looks like I'm just about right on time because I'm at my conclusions. So overall, the gene abundance patterns as well as the genome abundance patterns really do reflect the biogeochemical patterns both within and across sites. And methane flux is influenced by a really complex set of factors that include archaeal methanogen abundance and identity, the methane oxidizer abundance and activity, and a methylphosphonate breakdown. And it seems like site and salinity and biogeochemistry and microbial activities are really all important considerations when planning wetland restoration efforts and predicting greenhouse gas emissions. And what we'd really like, like I said, is develop a better predictive understanding of what happens before we restore these systems so that we can really maximize their potential to sequester greenhouse gases. So I think I just have one or two more slides, which I think, let's see. Just acknowledgments. Some really amazing postdocs have worked on this work, including Susie Thoreau, Wyatt Hartman, Xiaomei He, Jingli Zhu, and Cliff Bueno de Mesquita. Tiana Glavino Del Rio, some of you might know as a project manager at the JGI, who manages a lot of the metagenome projects and is just amazing at keeping things on track. Yi Tian was an intern who helped with some of the substrate incubation experiments. And then, like I said, Dennis Baldacchi, as well as Lisa Marie Windemeyers and Mark Waldrop of USGS were incredibly helpful in just getting this project off the ground back when we really didn't know anything about field work or how to study these sites. And so they were invaluable. And then the JGI Community Science Program supported a lot of that sequencing, and as did the DOE Early Career Research Program, which I was funded by for several years. And then I think the last slide is just a little JGI pitch, which I'm sure you've heard lots of, or maybe I left out the JGI pitch. But I can take questions. I was just going to note that, I don't have a JGI slide, but, the JGI does have some amazing user resources. And if people are interested in them, they should go check out the user calls there, in addition to EMSL, in addition to a joint call with EMSL, which is a really great opportunity to combine these capabilities in one proposal. So with that, I can take questions.
Susannah, thanks so much. That was great. Really interesting talk.
Thanks.
So I think we do have a couple of questions that are showing up in the Discord. So I'm going to go ahead and just kind of verbally ask those.
Okay.
So we have one from Paula. They are curious to ask about the relative contribution to methane production. And would you say that CP lyase contributed more than archaeal methanogenesis?
Sorry, I tried turning my camera on and it immediately started breaking up. So
Your camera betrayed you.
Like I said, that is really our big question that we'd love to have a clean and obvious answer to, which I think we're still trying to think about the best way to do that. I would say that the data from EMSL, particularly our struggles to find actual methylphosphonates at a sufficient abundance in these soils to fuel that constant methane production, has led us to think that the archaeal methanogenesis might ultimately be the more dominant pathway, but we still don't have like process measurements that would really tell us that. So we're still hoping to figure out the answer, but the jury's still out.
Okay. We have another question. I think there was a question about metabolite profiles from Becca and how well they aligned to K-base, but it seems like you did have a quick slide on that talked about that a little bit. So maybe we'll just table that one, and I'll move on to, have you seen marked stratification? So this person is mostly interested in how much methanotrophs can be decreasing the release of methane before it leaves the soil.
Yeah, I think, we definitely see differences in the communities with depth. I wouldn't be able to say offhand, but we haven't looked super deep, although I'd say that depth is kind of the least, pronounced effect that we usually see. But one thing I didn't talk about in the salt ponds is that although we do see an inverse correlation of, methane oxidizing genes or like PMO genes with methane, when you really look at them, they tend to look like they're probably from ammonia oxidizers, not methane oxidizers. They're hard to distinguish on a sequence level. But when they're in at the genome level, it seems like ammonia oxidizers are anti-correlated with methane, and like why that is, I'm not sure, but we aren't actually seeing a high abundance of methane oxidizers in the salt ponds, as far as we can tell.
Bram had a quick question. Are they taking up methyl phosphates so quickly that it keeps abundance low?
I mean, that is, that would be possible, except that I don't know why, like thermodynamically, I guess our assumption was that the methyl phosphates were made, at some other time or some other place, and then ended up in the sediment, because having simultaneous methyl phosphate production and breakdown doesn't seem like it would be energetically favorable to both processes. We don't see genes for, we also have looked for genes for methyl phosphate production and have not seen those in the unrestored salt ponds, although they are present in the historic salt marsh.
Okay. Okay, I think we have time for just one more. So one from Paula. Did you find any metabolic potential for methylamine utilization by the sulfate reducers in your databases, or your data sets, I should say?
I can't say we've actually looked in the sulfate reducer genes to see if they could take up methylamine. I think the general belief is that they can't take advantage of methylamine, but I'm not a metabolic expert on why it's thought that they can't, why that couldn't happen, or maybe it just wouldn't still be energetically favorable. But yeah, we have not looked in those genomes to see if those genes are present.
Okay, yeah, lots of metagenomic data to sift through, and lots of different questions that can be asked and answered with these large data sets, which I think is one of their great advantages, is that you can collect data and answer a bunch of different questions, maybe even ones you hadn't thought of yet.
Yeah, definitely.
Well, again, Susannah, thanks so much. That was a really great talk, and it gave us a really good view of how this kind of metagenomic data, once it's collected, and as you've been doing, sort of interpreted and visualized with all of the great slides that you showed us, how it could answer some really important questions related to this sort of carbon cycling. So again, I appreciate you very much.
All right. Thanks so much.